Oh.
Shit. Oh, what things?
A very good morning to everybody. All are welcome to the special occasion of international webinar on science, biology and the world's future organized by the Department of Botany in collaboration with IQAC of Dinobandhu Andrews College, Kolkata, West Bengal, India. This is our privilege that we have Dr. Bruce Alberts, the renowned scientist, present among us to deliver his valuable speech. Thank you, sir, for gracing the event. Now, I'd request our principal, Dr. Somnath Mukhopadhyay, to deliver welcome address. Sir.
Can't hear. I'm not hearing any sound.
Principal sir, if you can unmute yourself, please, sir.
I think his microphone is muted.
Yes, sir. Yes, sir. We're just about. Okay, sir. Sir,
Should I
sir.
start from beginning?
Yes, sir. Yes, sir. Yes, sir. We
Okay,
haven't heard
okay,
anything
okay.
from
Thank you, thank you.
No, sir.
Just a minute. First of all, I thank. Am I audible? Am I audible?
Yes, sir.
Yes, yes,
Yes,
sir.
sir. Yes, sir. Absolutely.
So I
You can,
start.
you can start,
First
sir.
of all, I thank the teachers of the Department of Botany and the members of Internal Quality Assurance Cell of Dinabandhu Andrews College to give me the opportunity to deliver the welcome address at the international webinar where Professor Bruce Alberts will speak. To the biologists of our era, Bruce Alberts is a name that needs no introduction. Actually, I am too small a person to say even a few words about the towering personality like Professor Bruce Alberts. I came to know the name of Professor Bruce Alberts through his epoch-making textbook titled Molecular Biology of the Cell, first published in 1983. The molecular biology of the cell actually redefined altogether the approach to the study of cell and molecular biology in a number of novel ways. The book is written in such a lucid style that even a stranger to biology will have no difficulty in following it from beginning to end. The book is equally useful to students, working scientists, and teachers. Illustrations used in the book are simple yet elegant. The text is supported by a thorough list of references, and the selection of references is made in such a way that they can be accessed in most of the libraries. I saw the book for the first time in 1985 when I was a final year graduate student of zoology at the University of Calcutta. Since then, many textbooks have been published on cell biology the world over. However, molecular biology of the cell still holds the prime position in terms of popularity among the general students, researchers, and teachers of the institutes of higher learning. For my part, I consider myself as one of the students of Professor Bruce Alberts, and I am fortunate for that. Incidentally, I had the opportunity of seeing Professor Bruce Alberts in The Living God of Molecular Biology on 13th October 2017 at the Presidency University, Kolkata. Seeing Professor Alberts face to face was not only a memorable experience, but also was like a transcendental experience for me. Most of the professors in the field of science believe that their prime goal in teaching is to produce more and more scientists. Professor Bruce Alberts, however, taught us that we need to have more people with science background in a host of other fields too, such as industry, journalism, government, and as teachers in schools and colleges. Echoing the philosophy of Professor Bruce Alberts, let us encourage our students to be open-minded about choosing their careers, wherein they can put their acquired scientific knowledge to its best use. Let us also urge academicians in the field of science to be more inclusive by welcoming also those who are using their science education to improve schools, businesses, and other institutions, along with the academic scientists and researchers, which constitute what is known as scientific community. Let disseminating science and its values be the academia's chief mission, a mission to which I believe Professor Alberts also subscribes. I sincerely hope that today's webinar is going to be a memorable experience for all of us. Thank you.
Thank you, sir. Thank you. Thank you, our principal, Dr. Somnath Mukhopadhyay. Now, I would request the convener of the program, Dr. Joy Sarkar, to formally introduce Dr. Bruce Alberts.
Thank you, Dr. Rupa Chakraborty. Hello, everyone. Professor Bruce Alberts, distinguished guests, and all the participants. A very good morning to all of you. Today, we have assembled here to witness a webinar organized by the Department of Botany in collaboration with Internal Quality Assurance Cell, Dinabandhu Andrews College, Kolkata, India, on the topic of science, biology, and the world's future. Scientists all around the world must now band together to help create more rational, scientifically based societies that find dogmatism intolerable. Today, we could not agree more to these inspirational words by Professor Bruce Alberts, one of his famous statements. Today, we are fortunate indeed to have Professor Bruce Alberts with us as the eminent speaker. Dr. Alberts, I am sure, needs no introduction. Dr. Bruce Michael Alberts, who is both a biochemist and a scientist with a strong commitment to improving science and mathematics for the greater good of humankind. However, since it is customary on such occasions to introduce the distinguished speaker to the audience, I have great pleasure in recalling a few of the numerous achievements of Dr. Alberts. Dr. Bruce Alberts took NAS Award, that means National Academy of Sciences Award, in molecular biology for the isolation of proteins for DNA replication and genetic recombination and the elucidation of how they interact with DNA in the year of 1975. He was awarded the National Medal of Science by former US President Barack Obama in 2014. Dr. Alberts served as editor-in-chief of Science magazine in the tenure of 2008 to 2013 and as one of the first 3 United States science envoys from 2009 to 2011. He is now the Chancellor's Leadership Chair in Biochemistry and Biophysics for Science and Education at the University of California, San Francisco, to which he returned after serving 2 6-year terms as the President of the National Academy of Sciences. As a humble student of the world of life science, I would like to mention with added emphasis that he is also noted as one of the original authors of the Molecular Biology of the Cell, a pathfinder book of all life science lovers. Unfortunately, it won't be possible for me to mention all his contributions in this short introduction, since we are in great pleasure to have with us such an eminent international scientist in our Bengal, though virtually. We are genuinely grateful to you, sir, for accepting our request to share your valuable time and giving us this golden opportunity to watch and listen to the observation in one of the prophets of the world of science. We have prepared a short video on Professor Alberts. With your permission, I would like to share the video on this platform. Ritu?
I'm going to give you a little bit of personal history because young people like to know how people like myself got into their career, and especially like to hear about all the mistakes that we made, because you're going to make mistakes too. So there'll be a little bit of that too. the idea of keeping pushing to develop the fundamental knowledge we need over the long run for innovation and understanding that will really create huge medical advances is, I think, being underappreciated. And we really have to make sure that the next generation have the chance and the incentives that will enable them to be innovators in developing the understandings we badly need. I had no exposure to science, really. My father was a mechanical engineer who became a patent attorney. And my main frustration was that he wanted me to help him fix everything. So instead of playing softball, which was my passion in those days, I had to, install yet more wiring in the baseboards of our house so we wouldn't have to have an extension cord anywhere, or something like that. So, but I did learn from him a lot about tinkering and making things work and solving problems. And I think, science is a career which I think is a great career where you get paid for solving problems.
National Medal of Science to Bruce Alberts, University of California, San Francisco, for intellectual leadership and experimental innovation in the field of DNA replication, and for unparalleled dedication to improving science education and promoting science-based public policy.
Thank you, sir. I now request Dr. Bruce Alberts to deliver his valuable words. Now the virtual platform is all yours, sir.
Well, thank you very much for that amazing introduction. I must say, I've never had a video prepared for any speech I've ever given. So this is the first, and it obviously took a lot of work and a lot of skill. You did a great job. I don't know if I need to give a talk. You know what I'm going to talk about. At any rate, it's a pleasure to be here virtually. And I just want to emphasize that there are some advantages of this new Zoom era when people can't travel, because I've been giving many talks. This is my third talk to India in the last month. And I'm talking to Brazil, Portugal, and all kinds of things that I would have had to travel for, and I would have done much less of it. And so thinking in the future, I think we've all learned that virtual meetings and much smaller events than this can be very productive. And for science itself, I think we need to think broadly about how we can make use of this ability to make science more productive and more, especially more collaborative. And that's a long subject that I'm not going to talk about, but I just want to emphasize that although I'm sorry I can't be there in person, it is a new era where we've, to our surprise, learned that Zoom kind of technologies really can be effective if used well. All right, so now I have to try to start sharing my screen. I gotta get rid of all the other things on my screen. Hold on. Okay. Should be better at this. I've done it enough. I should be better. All right. Get sharing it. Hold on. Oh, who can search? I'm getting controlled. Let's see, let me just try to shut that. Okay, there we go, finally. Can you all see that?
Yes, sir. Yes, sir.
Yes,
Yes,
sir.
sir.
Yes, sir.
Yes, sir.
It's very clear.
So please let me know if the slides stop showing, because that happens sometimes. But at any rate, I just want to start with a little outline. Of course, any talks everybody give all are based on their history and their experience, and so there's no exception here. Obviously, life is an education. I've learned a lot in a long life, and so I'm going to talk about 4 different things. From all my years of writing textbooks, I just want to emphasize how little we really understand and how many great opportunities and how much need we have for young people to do the kind of cell biology exploration that I'll talk about, to really enable us to reach better understandings. And the second thing I'm going to talk about is the fact that we need more fundamental research, not just translational research trying to attack known problems, because we know from past experience that that underlying knowledge is what will make possible really great advances in the future, even though we can't predict what they will be. Thirdly, because we have lots of students here, I would like to talk a little bit about the question of what should you do as a scientist. There's so many possibilities. It's a great career, many things to do, many problems to solve. How do you decide what to work on, and how do you decide, what exactly to do? All right, and then the last, of course, is my science education. I always end the talk talking about science education, and in this pandemic, in this crazy world we live in, it's never been more important. So I'm going to start with this issue. I've been writing this text. Actually, he was working on the 7th edition of the Molecular Biology of the Cell this afternoon. It's now evening in the United States, but it's been 42 years working on this. It all started quite accidentally when this famous man, Jim Watson, the co-discoverer of the DNA double helix, called me on the telephone and asked me to join him and 2 others as the author of a new textbook that he thought was really badly needed, a textbook that would bring together molecular biology, the chemistry of life, the field that I was working in and my background represented, and then a separate, at that time, very separate discipline called cell biology, which came out of the 18th century or 19th century microscopy and light microscopy, and the 19th century, and eventually electron microscopy also in the 20th century. And that's hard to believe, but cell biology was quite separate discipline from molecular biology at the time. And Watson had the really insightful vision that a new kind of textbook would be very useful to science if we could bring this off. So he had convinced us to work on this textbook by saying it would only take us 2 months, 1 month in the summer of 1978, and maybe 2 months in the summer of 1979, and it'd be all finished. And of course, this is the way great visionary leaders get people to do things. Because if you told them how hard it was going to be in the first place, nobody would start. Well, it was very much more than 3 months. We spent over 365 12-hour days working together as a group, these authors listed here. And at times we thought we would never finish and we should quit, but eventually we learned enough by writing, and we learned a tremendous amount by writing, that we finally made it work. And what kept us going during quite a depressing, hard slog trying to create this textbook was my co-author, Keith Roberts, who's both a plant cell biologist and a wonderful artist. And he designed all the figures, which are really, as somebody said, a unique feature of this textbook. And his general rule for figures, which is quite different from everybody else, is that if you can't understand it in 3 seconds, you can't understand what it's about, then it's too complicated. So there are many students who tell us they read each chapter in the textbook first by looking through all the figures in that chapter and then go back to reading the text. And it was so much relief and fun for the authors to work with Keith. We'd take him some ugly scribbling drawing and he would turn it into something really wonderful. We worked mostly in the UK because the other authors, except for me and Watson, were living in England. And this is a picture we took right around the corner from the house where we've been working. It's at the recording studio where the Beatles did their initial recordings, and they made the famous Abbey Road Crossing album cover. So this picture was taken as a joke, but then the grandson of one of the authors suggested we put it on the back cover. We almost didn't do it because we took a vote and it was, I think, 3-2 that we should do it. And it turned out that putting Beatles covers on the back of our textbooks, which we've been doing forever now, is probably the most popular feature of our book. This is the last edition, and I just want to use this to emphasize the fact that each time we write a new edition, we're humbled by how much we still don't know. Of course, when you write a textbook, you basically talk about all things we do know, and we don't want to give students the wrong impression that all the problems are solved. I think we would estimate that 10% of what we have to know about cells is what we know. It's easy to collect facts, collect huge amounts of data, but data is not understanding. So at the back of each chapter this time, we put a little feature, what we don't know, just some of the questions that were important to emphasize to students that there's a huge amount that needs to be researched in the future in this field. So I've been around for a long time, and I see the complete revolution in the way we understand cells. I came out of a physical chemistry laboratory, and we were trying to apply physical chemistry knowledge to understand DNA molecules at the time. And the way we thought of a cell was like a tiny test tube full of highly concentrated, disorganized individual proteins and RNA molecules freely diffusing and bouncing around. And that's because we knew how fast collisions, how fast molecules moved and twirled around their acids, the huge amount of motion at the microscopic level. And so that's how we thought everything worked. But of course, this was all wrong. An early discovery was the importance of so-called protein machines. And we now know that almost every process in the cell is driven by a complex of 10 or more proteins. And at least one of those proteins in that set is driven by allosteric changes that require energy, usually by ATP or GTP hydrolysis, but sometimes by gradients across membranes. They're driven to change their shape in basically one direction, and so those allosteric changes are like serve the role of the electricity in many of the machines we know about in our existence, which of course makes these machines make our life a lot easier. For the same fundamental reasons, these machines made life possible, because the chemistry that they could catalyze is so much more sophisticated than any other chemistry we know about. So this is a movie, you saw a little bit of it. This is my favorite movie because I worked on this problem for 30 years, and it was a complete mystery when we started. And if anybody had predicted it works this way when we started, we would probably thrown them out of the lab, because it seemed so crazy that we knew that both strands of the double helix went in opposite direction, but to think that one strand could be made continuously and the other strand could be made in a series of thousands of very short pieces that were then sewn together just seemed like a crazy idea. But it turns out by experiments, we eventually discovered by in vitro biochemistry that's in fact what happens. And this is just a movie of one particular protein machine, but there this is how all of life works. It's this very sophisticated chemistry. This is from our textbook.
Using computer animation based on molecular research, we are able to picture how DNA is replicated in living cells. You are looking at an assembly line of miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called a helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
So that gives you a real sense of how sophisticated the chemistry is that enables us to exist. It's really impressive. And that's why it's so hard to figure out. Okay, a recent breakthrough working in the same way that we had worked with purifying proteins, but we studied a very simple system, a bacteriophage system with 7 proteins. And now they've been able to reconstruct the whole process of replication and its initiation in a eukaryotic cell. It required 42 different polypeptides, and it's really impressive. And when you have such purified systems, you could actually figure out what's going on in ways you couldn't do otherwise. And so this is what they figured out in the cartoon version, how DNA synthesis begins, and then how the replication fork moves. This is forming 2 replication forks at a replication origin. They go off in opposite directions. Each one is more or less like the one I showed you in the movie. Now, it's humbling to think that all this complicated stuff is only a tiny bit about what we need to know about DNA replication and DNA repair. There are many more proteins involved, and DNA repair is an incredibly complex and important process that has many different pathways, and those machines need a lot more investigation and working out the details. And the reason is that understanding these molecular details of DNA replication and DNA repair pathways will be critical to improving human health. Of course, they're also very enjoyable. I mean, there's a huge amount of aesthetic value in watching that DNA helix replicate and seeing how beautiful the mechanisms are that make life possible. But they also have critical practical value. And the example I'll cite is that cancer research, which is obviously very important. So many people die from that disease. There's a field called personalized medicine in which we hope that eventually we'll be able to analyze the details of every individual person's tumor, because mostly they're all different. They arose by a chance process of mutation, multiple mutations, and figure out how to kill that tumor without killing the person and then get rid of the disease. And the basic idea is that if you actually knew understood enough about the biology, you could very selectively kill those tumor cells. And for example, the fact that for a cancer to arise, it has to accumulate multiple mutations in the clone of cells, maybe 5 or 10 different mutations that are selected for before the cell actually becomes dangerous. That causes a tumor progression often to select for hypermutability, that is a cell that mutates more often than the initial cell. And if once you knew what was causing that hypermutability, which DNA repair process exactly, which DNA replication or recombination process exactly, in principle, you could then kill that cell by making it even more unstable. And that dream has actually been realized for one type of cancer through what's so-called the poly-ADP ribose polymerase inhibitors, PARP inhibitors. These are FDA-approved drugs for treating cancer patients, but they only work in a very small fraction of patients with specific defects, such as the BRCA1 defect. And so that's sort of the example that proves the point, but we need many, many more such examples. So a crucial challenge for the next generation of biochemists is to obtain the information needed to accurately describe the mechanism of every type of protein machine in a cell. And of course, we do know the mechanism for the ribosome, for RNA polymerase, for DNA polymerase, the replication. We know a lot about many machines, but most of them we don't understand. We may know their parts and what the proteins are that make them up, but we don't know how they work. And so this knowledge will require that in vitro biochemistry be done to reconstruct the reaction in a pure system where you could manipulate all the components and actually decipher the mechanism and make a movie like the movie I showed you of that particular process. So there's now I want to shift to more recent realizations. You know, we wrote textbooks for years before we recognized either of these facts. The first is that the extensive sets of scaffolds, special protein and RNA molecules, produce biochemical subcompartments in the cell without requiring a membrane. Most recently, we recognize that many of these assembled by phase separation principles to create phase-separated liquid compartments. And I'll talk about that in a second. And then the other fundamental realization is that cell chemistry is so complicated that even when we know all the facts and have a network of all the reactions, we still won't be able to understand it because the real properties of life are so-called emergent properties that will require new mathematical and computational methods to decipher. And I'll very briefly talk about that. So scaffold proteins, this was a cartoon from our textbook of a scaffold protein. It's a protein that has a lot of flexible regions. It's generally placed in one specific part of a cell, and it binds these colored molecules that need to react with each other. And because they're held in close proximity, it greatly speeds up the collision rate between them, and therefore makes the formation of specific products much more efficient. Not only much more efficient, but creates those products in a specific part of the cell where they function. And this is the first example that was worked out in detail, as far as I know, from Tom Pollard's lab studying actin polymerization. Actin is a fundamental cytoskeletal polymer, very abundant protein. It's this red thing with a dotted line going through it. It would go down, you know, hundreds of feet through the basement of the down through the earth if you're drawing it correctly. This is just the growing end of a very long filament, and as shown here, there's a class of proteins called formins that often direct the polymerization of the actin, and they not only speed it up, but cause it to occur in specific places in specific ways. And the biochemists studying this noticed that the rate of growth of this end was faster than so-called diffusion control. Diffusion control comes out of physical chemistry, where you can calculate the collision rates of the actin monomer with the end of the actin filament, and to grow faster than a collision rate is impossible. So the only reason it works is that you have these scaffolds or whiskers that are on the formin that serve as loading zones for the actin monomer, as illustrated here, so that the rate of collision is much faster, and therefore the growth rate is much greater. This is the simplest example I know of. Most of them are much more complicated. RNA scaffolds are also widely used in cell biology. They're more recently recognized, so-called long non-coding RNAs. Some of them have that role. we've actually known about the idea for a long time, but you know, we didn't really understand what was going on. Here's a 1994 light micrograph with a stained cell nucleus showing that where you make RNA molecules, the transcription factory, you aggregate them into these special places called transcription factories. And when you do DNA replication, you make these green areas called replication factories. Those are really a way of greatly speeding up both those reactions by bringing those many hundreds of molecules that need, proteins and RNA molecules that need to interact with each other into close proximity, so those reactions occur more efficiently. And we never really understood those until more recently with this phase separation intercellular condensate idea based on forming liquid-liquid separation of components through weak fluctuating interactions. Here's we now call these things intracellular condensates, and they serve to form many different membranous organelles in a cell. This is just a simple diagram again from textbook. But we didn't understand that at all. We still have a lot of work to do and figure out how these condensates form and be able to understand what limits their size and so on. So, but in conclusion, a cell is nothing like a test tube. Nearly everything is organized inside the cell by protein and RNA scaffolds to make the chemistry of the cell most efficient. And it needs to be that efficient to really give rise to the kind of life that we know about. So the second sort of failure we had in early days to understand what we needed to know about cell biology was the whole idea of so-called emergent properties. This comes out of physics originally. In 1940, there was a famous paper on emergent properties. When you get any system that gets complicated enough, it has properties that are more than the sum of its parts. And this is, of course, how nearly all of biology works. We all learned when I was a student, A goes to B, goes to C, goes to D, goes to E. But now we understand there's all these feed forward and feedback loops that are needed to control those reactions, so they're really useful for a cell. And even this kind of simple diagram can't be understood without mathematics. Well, here's a moderately complicated example that involves that same actin molecule. This is the different molecules that control what actin does, the so-called actin binding proteins. And these are just from a yeast cell. A human cell would be even more complicated. We have the filament dynamic molecules like the formin that I showed you in that a few slides ago. Those are the dark purple ones. The famous molecules, myosins, are green. Those are the motor proteins that move along actin filaments. Anyway, you could now, with all this technology, we have the structures for all these proteins. We can get them. We could find out their interaction parameters with binding constants, their on and off rates in different complexes. But the whole thing is so complicated, we can't actually use that information in our heads or yet with computers to really figure out what it all means and what it all does in the cell, to organize the actin in many different ways and give it the properties that it needs to have for our cells to function appropriately. So as a consequence of such complexity, which is everywhere, cell signaling is a network of very similar interactions. Even when we gain a complete knowledge of all the molecules, protein machines, and molecular interaction cells in a cell, we will not be able to truly understand it. This is true even of the simplest of living cells. Instead, life reflects the emergent properties that result from very complex networks of interaction. So this is my conclusion. We're going to need a lot of work from the students that are online. Much more biochemistry and purified systems, also new quantitative methods for analyzing and understanding the enormous complexity of life's chemistry, we don't even know what these are. They're certainly going to involve computer science and mathematics and engineering, but nobody's been able to really come to grips with this in an effective way yet. Okay, so because of all these unknowns, we have a lot to study, but why is that important? And in this country, we need to convince politicians that fundamental science is important. And of course, politicians are driven by short-term interests. And they want us to improve human health if we're or human agriculture or progress in many other applied fields. But what history shows, and it's very clear that, you know, the best way to really improve humanity is to invest in fundamental knowledge, which then in unpredictable ways will allow us to really improve our environment, improve our health, improve our food and everything else. So the current problem in the United States is that the major funder of biological research is the National Institutes of Health. And under pressure from disease groups, politicians, and others, in my opinion, they've been overemphasizing translational biomedical research, which is important, but it's not all that's important. We have to do more than the short-term stuff. We need to do the long-term. With so many unknowns and so little understanding, fundamental research on biological mechanisms also remains absolutely crucial for improving human health over the long term. And I was editor of Science magazine. For 5 years, I published 250 editorials. Most of them were recruited from outside people. Of those 250 editorials, this is really, I think, my favorite one. This is from Huda Zogby, a great translational researcher. She started as an MD. She's at Baylor Medical School. She was treating children with Rett syndrome and got frustrated by the fact that she couldn't do anything for them. So she became a researcher and she became a very distinguished one. Still, she's trying to solve fundamental disease problems, but she has the background and the experience and the prestige to be able to say that today, she says, many highly qualified basic scientists feel compelled to jump on the translational medicine bandwagon. And I've seen that a lot among the young scientists that I interact with. And the favorite part of this editorial is what she came up with in this little quote at the top. This is from her editorial. She's from Lebanon. She, I guess, grew up speaking Arabic, so she knows the difficulty of language. And she says that, you know, people trying to do her job of curing disease are trying to translate, you know, a text with the sophistication and depth of Shakespeare using a first grader's vocabulary and experience. That the first grader's vocabulary and experience is what we now know about cell biology. All that stuff you could read in our textbook is only what she would call a first grader's vocabulary and experience, and I would agree with that. So here's an example of what I've been talking about. This is a paper published by a group working very hard with clever methods to make the smallest living cell possible. They started with this tiny bacteria called a mycoplasma, and using synthetic DNA methods, they were able to select for the smallest number of genes that would enable this organism to still double in 3 hours. You know, so it would still grow at a reasonable rate. And to everybody's surprise, including theirs, when they did that, out of the 473 genes on their list, they had no idea what 149 of them did. And that just shows us how ignorant we are of even the simplest biological cell. There's very few people trying to solve these problems. You know, it's not fashionable to try to work out what these 149 proteins are doing. But I'm very sure there's several Nobel Prizes hidden in figuring that out. I mean, it's fundamental, but it's not fashionable to work on these fundamental issues. Another example is the importance of Drosophila, the fruit fly. We're still very far from understanding how cells work together to form and maintain tissues in a multicellular organism. The cell has to do a lot of interaction with its neighbors, know how old it is, and then decide what to produce and what to do, whether it should kill itself, whether it should differentiate, whether to just sit there and not do anything. So in some sense, the signaling networks in cells are allowing the cell to think in some way. But many examples show that first working on a mechanism in Drosophila provides a shortcut to understanding humans. And I use as an evidence our textbook because our chapter on the development of tissues has 50 references to Drosophila, which is 4 times more than the next most cited organism, which is the mouse. It's just been astounding how much we've learned from these model organisms, including other ones, of course, like the worm C. elegans. So consider the human brain. This is, many people say the ultimate emergent property is human consciousness. It comes out of a network of interactions of cells and very complex synaptic networks. And this completely different thing, human consciousness comes out of that. That's a really hard problem to work on. So hard because the human brain contains more than 100 billion neurons, each of which on average has to make connections with 1,000 others according to a regular and predictable wiring plan. This is just astounding. The human brain, just the most amazing thing that I could think of. But, the Drosophila brain, which has got the same kind of mechanisms, it's so much easier to study. We're never going to understand the human brain until we can understand the Drosophila brain. And that could be the foundation for much greater learning. But again, relatively few resources are put into this. It contains about 100,000 neurons, 1 million times less than humans. So it's 1 million times more less complicated. Also, of course, you could trace every cell, you could mutate any gene, all the kinds of things you can't do in a human. This is where we're going to really come to grips with a lot of basic knowledge that we need to understand the human brain. And yet, the funding agencies tend to push this kind of research into relatively poorly supported research areas. So we also need new models. I'm just advertising one of our young professors, Wallace Marshall, who had the courage, very, very rare, to shift what he was working on to come back and try to tackle a really important problem nobody else was dealing with. It's been around for more than 100 years. We've known that this giant ciliate cells like stentor, which T.H. Morgan worked on before he worked on Drosophila, and other people worked on until about 1950, and then people just stopped because they didn't have technology to make progress. But now we do have that technology. But it's a giant single cell with an intricately patterned plasma membrane. It's like the single cell has a GPS system where it knows what's supposed to be in every part of its surface. And this is a great place now with new technology to try to work out how that happens. And I'm very sure that once we understand that, we'll have many applications to understanding human biology. And we don't even notice this feature of human cells at the moment. And then we have a new model organism, thanks to this enormous tragedy that we're all living through. Coronavirus has been a disaster for my country. Had very poor leadership. We're very unhappy about the inadequate application of science at the national level in the United States. But it's causing enormous tragedy, but it's also stimulating a huge amount of very high quality cooperative research on how it works across the globe. And this is a really interesting problem because this is a huge virus. It's 4 times larger than the flu virus. It makes 29 proteins, most with functions we do not understand. there had to be something in the evolution of this virus that made it advantageous to get all these many proteins. It has many disadvantages. For example, it has to make RNA by a more accurate mechanism than other viruses do. It had to get a whole proofreading mechanism selected for. So there's just a deep failure of anybody to understand this virus and why it exists in such a complicated form. And the massive mobilization of scientists now is going to make understanding this one model organism, really unique. And we're going to gain huge amounts of new fundamental understandings, both about viruses, but also about human cells. I'll just give you one example. This is a paper published largely from UCSF involving more than 100 authors, an enormous cooperative effort involving labs across the globe as well, where each of the coronaviruses proteins was used as an affinity tag to identify the human proteins that it interacts with as it's infecting a cell. And these diagrams are just a summary of, each red dot interacts with a whole group of different kind of proteins and different systems. And to me, this is a striking illustration of how little we understand about both the basic cell and about the immune system, which is targeted by many of these proteins in order to help the virus escape from the immune attack. And that's presumably part of the reason, at least, to why it's so complicated. But again, we just don't know enough to be able to exploit this knowledge yet, make effective drugs. This is a part of a drug search effort, but we're going to learn a lot about just fundamental cell biology from all this wonderful work. So I just want to mention another problem in the US today. I hope it's not a problem in India, but in the United States, when I was becoming a young scientist, most of us got our own research groups as assistant professors at age 28, something like that. Shortly, we got a PhD. We went for a 1 to 3 year postdoc, and then had our own independent labs. But look what's happened. In 1980, there were 2,500 independent labs headed by scientists age 36 years and younger, but we've lost more than 2,000 of those. What's happening instead is older and older people more of us are running labs. So the fact that proportion of all grant funding awarded to scientists under the age of 36 fell from 5.6% in 1980 to only a little over 1% in 2012. This is astounding. So this is a question I would like. It's obviously got an easy answer, but it really makes you think. How successful would Silicon Valley be if nearly 99% of all investments went to scientists and engineers who were 36 years old or older? And in addition, another feature of our system, there was a strong bias towards funding non-risky projects, which had a high probability of working. So, in fact, Silicon Valley wouldn't exist. We wouldn't have smartphones. We wouldn't have a lot of other things. So my colleagues and I, Tony Hyman, Krista Pickett, Shirley Tillman, and Harold Varmus, published this paper proposing how to solve this problem, which we're still pushing our government to do. We have not been completely successful yet, but we're getting some attention. We'd like for young people to have an equal chance, an equal number of grants awarded for the standard kind of grant, which is continuing whatever you did before for postdoc, and the different kind, which is called a new innovator grant, where you could propose anything. You don't have to have done it before. It's going to be riskier, but this is where innovation is going to come from, and we want the NIH to make it possible for young people to start working on their own ideas in new labs earlier. Okay, so now I'm going to talk about specifically to younger scientists, how to decide what to investigate as a scientist and how to proceed with your research. And I'm going to do this by talking about a course we've been teaching at UCSF, and it's called a mini course. UCSF has these mini courses where they're all over in 2 or 3 weeks. They don't last for a whole semester. 15 students meet with 2 faculty in 10 3-hour sessions over the course of 3 weeks or so. The students read and discuss a series of research publications, and they're offered 40 different topics every year. Any 2 faculty can make up any course they want to: evolution, aging, protein quality control, and so on. The mini course that I've been teaching with a colleague is called Classic Papers in Biology. I originally taught this with Cynthia Canyon, who then went off to a biotech company, and now I'm teaching with a younger faculty member, Denitsa. And it's called Classic Papers in Biology because we learn about great experiments in the history of science, but those are always paired with a more modern paper. And the course really aims to stimulate students to think innovatively in planning their thesis research. That is, we want them to think as a scientist like Mendel or Watson and Crick or Yamanaka or Darwin. So we've started with Gregor Mendel. It's a wonderful translation of his original papers, and on the fundamentals of heredity. And then we pair that with a new paper on an epigenetic inheritance in a plant. They also read, many of our sessions focus on Nobel Prizes or contain a Nobel Prize winner. We read the 2 papers cited by the Nobel Committee for the discovery that cells can be reprogrammed to become pluripotent. These are by John Gurdon and Shinya Yamanaka. And then I would like to advertise this wonderful talk that Yamanaka gave before he got the Nobel Prize. It's available on YouTube, where he describes how he devises a successful research strategy. And it's got just wonderful lessons about how to think about science. We do, of course, biochemistry. We start with this Nobel Prize winning discovery of DNA polymerase enzyme by Arthur Kornberg, and then that's paired with a recent single molecule assay. Amazing that you could actually look at what single molecules do using, this is the case, using laser forceps and very sophisticated technology. We then have 4 other pairs of papers. They change from year to year, but we always end with a session called Darwin and Scientific Standards. And we read 2 chapters in The Origin of Species, and then we pair that Darwin reading with a recent high-profile multi-author paper that has been shown to be completely wrong. And the discussion is how to avoid being misled, fooling yourself as a scientist. Because, it's very easy to do that for young people to get in an environment where there's only one right answer for their research. And that's the point we try to make. But why do we pair it with Darwin? Well, Darwin was really a model scientist. If you read Origin of the Species, you see he's always trying to think of what could be wrong with what I'm saying. And what experiment can I do to test this seeming inconsistency? And this is from his autobiography. He said, I had also during many years followed the golden rule, namely whenever a published fact, new observation or thought came across me, which was opposed to my general results, to make a memorandum of it without failing it, and at once, that is to write it down. For I had found by experience that such facts and thoughts were far more apt to escape from the memory than favorable ones. This is just the nature of us all. We tend to think about the things, remember the things that support our ideas and not the things that that conflict with them. And it often leads scientists to make terrible mistakes, find drug targets that don't exist, and so on. So this is me. This is the first year we taught it by Zoom. So this is the class, pictures of, I actually had pictures. I visited Darwin's home, Downhouse in England. I had pictures of his writing desk, which I said was a great inspiration. It should be a great inspiration to us all, but certainly is an inspiration to me. So our major point is that strategy is everything in science. Because as students discuss each paper, we dissect the experimental strategies that have been designed by each scientist to attack an important biological mystery. So it's first of all to work on something that's an important biological mystery that will make a difference, but secondly then to design a really effective strategy. And we point out how these strategies often involve an investment in developing a new method. Okay, I just want to emphasize that I've learned a lot from India, especially about how science can apply very broadly to even the most underprivileged parts of society to help with improving livelihoods. I've been maybe 20 times in India, I don't know, something like that. And my colleague there initially was MS Swaminathan, who still going strong at age 95. And one of my first visits in 1998, early visits, was to see his so-called information villages, where they were connecting scientists by wireless internet to groups of farming villages. And they just did this beautifully. In each village, they developed a core, usually of women who had never had more than a high school education, to run all the computers. And just incredibly impressive how they were supplying knowledge about farming practices. You know, people would come and take their diseased plant, hold it up to the computer screen, and get scientists to try to help them. And on subsequent visits, I discovered that this was connected to your self-help group funding mechanism. Here I am with MS Swaminathan next to me, with a bunch of bankers and the governor of Pondicherry. And here's one of the things we saw, the way that scientists had enabled groups of women to make a business out of selling biopesticides, and also, of course, to use them in their own farming practices to avoid the use of costly and often dangerous chemicals. Then I went to, when I was editor-in-chief of science, I made a special trip, I think it was like 2 weeks, with the science news staff to see how the major universities and IITs were trying to bring science to throughout India. And we published this wonderful long article in May of 2013 called Science for All. And it had a picture of an Indian woman scientist on the cover. I mean, Indian woman farmer on the cover, standing in her field. But it was so impressive to learn all the things that could be done with science and technology if you focus on the real problems of society. So I just want to end with my science education theme. And my first trip to India in 1993, I learned about the word scientific temper, which of course was invented by your first Prime Minister, Nehru, and he wrote about it a lot in the 1950s. And spreading a scientific temper throughout society will be essential and require that we change what we now mean by the term, quote, science education. So this is just the take-home lesson from Nehru's emphasis on science for India, that every nation also needs needs the creativity, rationality, openness, and tolerance that are inherent to science. He talked a lot about the fact that you had so many religions, so many different languages. You need a scientific temper to make your new democracy work. And of course, that's been very important for India. These are the values of science. I like to talk about honesty. If you're not honest, you can't be a scientist. You're not, you put out your results for other people to learn and they build on it. And if you're wrong, it's not good. Generosity to make the thing work. It's got to be a community effort. And very importantly, a strong demand for evidence with openness to all ideas and opinions, irrespective of their source. You always have to, if a graduate student has an idea that's different from a Nobel Prize winner's idea about some scientific problem that you're working on, you better pay attention, because it doesn't make any difference who says something. It's the idea itself that you have to listen to. And that's, of course, very important in any democratic society. So this is what I learn when I go around the United States, how far we are from teaching science the way we should teach science. This is what I learned when I gave a talk at one of our major universities. The mother came and told me her 8-year-old just came home from school and said, "Now I get it. Science is just like spelling. You just need to memorize it, and it doesn't make any sense." This is such a sad statement. But it reflects the way we often teach science as a bunch of facts and words to memorize. And in the United States, there's so many American politicians who don't understand science, hate science, are ignoring science, probably because this is the kind of science education they got. So we need to do much better. Otherwise, our democracies can't really be effective. Right now, we have crazy people in the United States spreading on the internet the idea that, oh, masks are not important. In fact, masks cause disease. They don't prevent you from getting disease. This is just contributing to the major disaster we're having. People not used to thinking about what science says and appreciating what science is and why they should pay attention. So this is the kind of a science that I'd like to see spread throughout the world. This is actually what the students in San Francisco were doing when I left in 1993 to go to the academy. This was the official curriculum. We had a great project with UCSF and the school district in San Francisco, and they did actual science. This is the first year of school. 5-year students put on clean white socks and walk around the schoolyard at a time when there's seeds on the ground. And they come back and their socks are dirty. They have a little forceps and a piece of paper with numbered white squares in it. And they pick out each speck, put in a different numbered white square. Then they look at each speck with a tiny little plastic microscope, it costs $3, and draw a picture of it. And then they have a debate in class, which ones are seeds and which are dirt. Well, since seeds evolved to stick to animal fur, many of the seeds will stick to the socks, but of course, most of the stuff will just be rocks and dirt. And so that this is taught well, the teacher won't tell them the answer. They wait till some 5-year has the idea that, well, maybe the regular spaced specks that we have from our socks, maybe the regular shaped ones are the seeds and the other ones are dirt. And of course, the teacher says, well, maybe that's possible. Let's discuss it. And eventually the class says, well, maybe that's possible. And then the next day they might have a discussion about how would we test our idea that we just discussed yesterday. And eventually the teacher waits till some kid gets the idea that you could plant all the regular shaped ones in one pot and plant the other ones that are not regular shaped in a different pot, and see if we only get plants to grow from the pot that has the regular spaced specks. So this shows that even 5-year-olds could do scientific investigations. And of course, we have designed over the course of the years many such challenges for kids that get higher, more difficult as they age. This is a group of 12-year-olds in San Francisco doing the kind of science that we helped to create at UCSF. It was called a project called City Science. The teacher is walking around the room like a coach. The class is noisy. The kids are trying to work in groups to figure out things that are more complicated than which are seeds or which are rocks, but just appropriate for their own age. So imagine an education that involves solving hundreds of such challenges over the course of 13 years of school. I think the children will become adults who would be great problem solvers in the workplace. Every company wants to hire people who could solve problems using logic and evidence. People who will be able to make wise judgments for their family, their community, and their nation. And adults who reject the kind of magical thinking we're seeing in the United States now. Some people, even our president said, "Oh, I think the virus is going to go away after the election," or there's crazy things that don't make any sense and which interfere with the public doing the right thing to protect themselves and protect our society. So we're heading up to 200,000 deaths in our country pretty soon. It's just tragic. So this is something that my friends don't like to hear. Professors don't want to hear this, but one of the things I really learned in my years at the Academy working on science education is that to remove a major barrier to progress at the early levels, pre-college levels, science education at the college level must change. And this is because the way we teach science in, say, biology, first biology class in college, that defines what science education is in biology, and likewise for physics, and likewise for chemistry. People will define science education. The standard will be set by the universities. And of course, the universities teach the people who become teachers, and people who are teachers will tend to teach the way they were taught. And so there is a revolution going on. It's a slow revolution. This is the University of Minnesota, where all 3,000 students who take Biology 1 take it in groups of 200, sitting around tables solving problems like the students here. We estimate now that something like 1/5 of all college science courses in the United States are taught in this active mode. So we still have not made the kind of progress we need. We need all science to be taught this way. The Academy published a wonderful book designed for faculty called Reaching Students: What Research Says About Effective Science Teaching in Undergraduate Years. And it's like everything else on the Academy website. It's free PDF can be downloaded from the Academy, National Academy Press website. And it's really an effective tool for getting professors, if they will start to read it, to think about what they're doing. It points out that nobody changes their lectures all in 1 year, and they're just urging college faculty to try 1 week of teaching differently in the first year. And it also points out all the research that shows that this form of teaching really is much more effective in getting students to both understand science and to understand what science is, which is critical now that we have so many democracies where social media is spreading just destructive, crazy ideas that are harming people's health. So this is really my favorite quote about science. It's from a little book written by the physicist Jacob Bronowski called Science and Human Values. Bronowski is a young man in the British Army, flew over Hiroshima and Nagasaki and was very depressed about what he saw. So it's horrific amounts of death. Did science create bad things for the world? He spent 10 years reading and thinking about the history of science and what it had done for the world. And of course, he concluded that science on balance was very, very beneficial for the world. He said the Society of Scientists is simple because it has a directing purpose to explore the truth. Nevertheless, it has to solve the problem of every society, which is to find a compromise between the individual and the group. It must encourage the single scientist to be independent and the body of scientists to be tolerant. Science has humanized our values. Men and women have asked for freedom, justice, and respect precisely as the scientific spirit has spread among them. This is really just a restatement of Nehru's scientific temper ideas. And it's just it's very motivating about for all of us who really think about our societies and the need to spread science much more effectively throughout our society. So this is the slide I used to show at the end of my talks about academy presidents working together across the world. The fact is that even when our governments don't speak to each other, scientists generally can share a common culture, can agree with each other. We're all unhappy that our governments make short-term decisions and don't listen to science enough. This is a picture taken in Swiss McDonald's after 15 academy presidents had worked for 3 days at Davos. And in this particular picture, we have the president of the Chinese Academy of Science, Japanese Academy of Science, Swedish Academy of Science, and two presidents, Prakash Tandon and Govardhan Mehta of the Indian National Academy of Sciences. So I've had a lot of experience with these people. We could work together really well, but we're not part of the world anymore. The world exists on social media. I don't think any of us are on social media. So I've been pushing the new development of so-called young academies. Young academies were first formed in Germany. It was a brilliant invention. And the Germans also support something called the Global Young Academy that works to support this new young academy idea. And there are now 40 nations with young academies. This is pictures I had to have of the members at one point of two different young academies. Here's how young academies, as invented by the German academies, differ from traditional academies. First of all, election is temporary for 5 years only. Typically, the young means 35 to 40, and they seek future leaders for these posts. So every year, 10 to 15 come in and 10 to 15 go out. So the whole academy is not very large. They could all interact with each other. Very importantly, unlike the major academies, accepting membership requires an obligation to spend time on public service. And there's generally at least one major Young Academy project each year. And the Young Academy is directed by an executive committee of some of its members. And thirdly, I think very importantly, membership is broader than a typical science academy. It includes not only engineers and social scientists, but also mostly, in most academies, political scientists, entrepreneurs, science communication experts, often a lawyer, somebody who would passionate about science and help connect science to other parts of society. And this is the only picture I could find of the Indian Young Academy. It's relatively new. These are the founding members. I think Indian Young Academy needs a better website. Anandita, who's not very far from you, in Kolkata, north of Kolkata. I've actually been at this, visited that ICER when it was very new. She's Anandita Badra's just been elected the co-chair of the Global Young Academy. So India is very well attached to all these movements. So this is my last slide. To create a scientific temper for the world, the challenges ahead for scientists are enormous, but by working together across the globe, we must find a way. This is my last visit, most recent visit to India in December of 2019. And here I am with a bunch of Indian students learning how to use these so-called foldscopes that I had brought a package of them with me to India. So thank you very much. Try to get this thing. Trying to undo my.
Dr.
Thank you, sir. Thank you, sir. Science has humanized our values. We have to remember that. Thank you, Dr. Bruce Alberts. This is really a memorable event for us that we attended this session. We have no words to express our feelings that we got the chance to enlighten ourselves with this speech. Now, this is the time for question answer session. It will be conducted by the senior most teacher of the Department of Botany, Associate Professor, Professor Subhashi Saha. He will conduct this session along with our esteemed faculty member of the Department of Botany, Dr. Krishna Choudhury.
I'm trying to stop sharing my screen, but I can't seem to be able to do it.
No problem,
What's going
sir.
on here?
No problem. You take your
Hello,
time.
sir.
Can you stop sharing my screen? I don't seem to have the control here anymore.
I'll request our convener.
Okay, good. All right. Is it okay? Can you see me?
Yes, sir.
Okay, good. Okay.
No, Bondo.
This is sir. Start questions.
Hello, sir. Hello, sir. I'm here to ask you a couple of questions on behalf of the viewers. Although we have received a lot of questions, sir, you please enlighten us. The first question is, all the funding houses are ready to fund mostly applied research nowadays. People doing basic research are striving in many countries around the globe. Many of us believe that most of the fundamental facts of the cells are still unknown. What is your opinion on this, and how do these researchers thrive in this scenario? Or what is your motivational words for them, sir?
Well, this is always a problem in every society, and I talked about it. We're vastly underinvesting in fundamental research. And of course, fundamental research is also a way to create much more cooperative science around the world, because when you're working on a new drug or something, it's often very competitive because there's intellectual property and possibly money involved. But fundamental science is the same everywhere, and we should be working closely together. I think the new Zoom culture is making some of us rethink about how we could really do a better job of connecting fundamental scientists around the globe. For example, I've been interacting with one of the young new faculty members at UCSF, who just arrived, and I think would greatly benefit from connecting to the 2 or 3 experts from around the world who could serve as advisors or mentors for him. I would like to see us try to develop a culture, now that we have this new technology, of students being able to choose from a large list of scientists. There are many scientists who reach an age where they shouldn't have a lab anymore, but they could be very good mentors to other people. And we're just toying with the idea of trying to create a list of people who volunteered to mentor other scientists, and then have young scientists choose people who they'd like to try being mentored by. In other words, you don't have, your thesis committee need not be only the people that are in your same department, but could include people around the world who have something to offer that you think has something to offer in the way of advice and connections. And I think this way this is a way of strengthening fundamental science. And that's happening on the coronavirus because everybody's, sees it that this is an emergency and collaboration is critical. And so we need to be able to continue reinventing science to make it more collaborative in that and other ways. The other question about how do you get governments to understand this? Well, my academy, when I was in Washington in 1995 or so, there was a threat from the new president, President Clinton. They floated a rumor that to get a research grant, somebody had the idea that you should have to explain in your research grant application, what if you solve that problem, what difference would that make for human beings? And we thought this was a terrible idea because it would make it very hard to support, or even harder to support fundamental science. So the Academy had a 10-year or 8-year project to produce a series of pamphlets called, they were designed for the public and for the politicians in the United States. And eventually I think they were part of the reason why that idea never took hold in the Clinton administration. There's a series of 8-page pamphlets which started with something, and the first one we produced was the Global Positioning System, GPS. And everybody knew that was really important for lots of things, changing lots of things, allowing you to do things you couldn't do before, knowing exactly where you were in the world. But we traced it back to where did it come from? And it turns out we started with atomic clocks, which were a great discovery by physicists 40 years before the GPS system was invented. We showed how in a central timeline in that Beyond Discovery pamphlet, how in unexpected ways, the discovery of atomic clocks was combined with something else, combined with somebody else, and over 30 or 40 years, and led to this amazing fact that you could have 24 satellites going around the Earth, each with an atomic clock in them, and that allowed them to measure exactly where you were. So enjoyed enabled the person on Earth to determine exactly where they were. But we did that 20 times. We did the cure for AIDS, HIV. We did the cure for AIDS. We did the cure for childhood leukemia. We did many different pamphlets. They're all 20 of them are still up on the web. If you Google Beyond Discovery Academy or Beyond Discovery National Academy, you can find them as free PDFs on the web. But I think every country has to do that in your, you could take that and other things like that and produce them for your government. We printed a bunch of these pamphlets. We made a 1-page summary. We distributed in every office of every politician in Washington. But that is also necessary. We need to get funding for this, and we also need to make the enterprise as efficient as and effective as possible by trying to think about new ways to be more cooperative in international science.
Thank you, sir, for sharing your ideas. Now the second question, sir. When you discovered DNA replication, what was the first thing that came in your mind about DNA replication?
Well, I've written about this. As a graduate student, there's a little thing about failure in my video clip. But I published there's a website that my colleague developed called iBiology, which I would highly recommend to students. It's got about 400 lectures by different well-known scientists about their research. But a few other things, I gave a 10-minute talk about the importance of failure. And in that talk, and also in an article I had published in Nature, a little one-page article, I talked about the fact that in my PhD research, I made a theory about how DNA replicates, and my professor let me spend my 4 or 5 years trying to prove that theory. Of course, the theory was wrong. It assumed that DNA was replicated by only one protein, DNA polymerase. This is what people thought in those days. And it ended up, the net result of all that was the first time I went for my thesis exam when I was about to leave from my postdoc, I failed it. And so that's the sort of the start of my talk on the importance of failure. But basically, you know, I thought DNA replication was an important, interesting problem. It was an important mystery. And it was just intriguing to try to work it out. So I first made, and I got an idea, which, you know, I showed it to Jim Watson. He was then a professor at Harvard, where I was a student. He thought it was a nice idea. He gave me encouragement. And then, you know, I did all these experiments, but of course, the whole hypothesis was wrong, that I didn't know anything about protein machines, and nobody imagined that there was a protein machine. But then after I finally passed my PhD and went to my postdoc, I did the same kind of thing that Yamanaka did. That failure made me think very hard, what kind of strategy would make sense? Since we knew so little about DNA replication, I should do something that would advance knowledge, no matter whether my theory was right or not. And so I invented DNA affinity chromatography. I made a column out of DNA hooked to cellulose. We call it DNA cellulose. And that was 1967 or so. And with that column and an extract of infected bacteriophage infected E. coli cells, I was able to isolate a new kind of protein that was needed for DNA replication that nobody had ever thought of before. And that sort of started me on my way of using a very different strategy than had been used before to try to get at that problem. Rather than assuming that I knew that only one enzyme was involved, I was going to try to use the genetics of bacteriophage T4 to see what actually was going on. And but I think it was important that I really was interested in the idea. I mean, I was just intrigued with the question of how DNA replicates, and it clearly was going to be an important problem to answer because it had so many implications. And so for young students, I talk about the fact that you have to be passionate about what you're working on. You really have to really care about the answer. Otherwise, it's too hard. And, you know, you have to really be working on something that excites you. And so that's the first requirement. You got to work on something that excites you. The second thing is you should really be careful to choose something that makes a difference. Once you solve the problem, it will make a difference for other people because it's an important mystery. It's not just some crazy mystery that nobody cares about. It's not going to be important to solve. And thirdly, and very importantly, that's the whole point of our course, you got to think very deeply about strategy. Try to learn from all these other people. Like I recommend the students to watch these iBiology videos, see what strategies other people who've been successful have used in their science, and always try to think about how can I attack my problem in a way that nobody else is attacking it. That's exactly what Yamanaka does in his little talk. He says, clearly, I want to reprogram cells, and there are a lot of people doing the same thing I'm doing. It's an obvious mystery, and it'd be very important if we could reprogram cells, take a fibroblast and make it into a pluripotent cell. But here's what other people are doing, and I can't do that because by the time I do it, everybody else will have done it. So I'm going to do it something else. And he makes an investment in a strategy that actually paid off. So that's the kind of thing that we like students to think about. you know, it's too easy just to collect data now. I mean, it's easy, there's some kind of, we could collect data on anything you want, but is that going to lead us to something important? And secondly, I would ask, are other people doing that already? That, well, why should I do it if it's already being done? I need to do something that's different, at least partly different from other people, so I have a chance of making a real contribution.
Thank you, sir. I'm sure it will help us to understand the concept more clear. And now it is over to Professor Krishna Chaudhary, who will ask you certain questions to you.
Thank you very much, sir, for your valuable talk. We have many questions, but a couple of questions we have selected for you. The first question is, how do model organisms arise in this world of science? Because in your journey of biology, we work with many organisms, all are never model organisms. And what's your opinion on this? How can young brains like us classify between potential model organism and non-potential model organisms for the research values?
You know, I would really, so you know, one thing that would be really interesting for young Indian scientists to do is to try to find some friends who want to work on the same system, and then you know, create a little nucleus of people who are supporting each other. I mean, I talked about the work that Wallace Marshall started working on Stentor, the ciliate. So there was a huge amount of background information that he was using from 100 years of work on Stentor by other people with microscopy, and people had cut it up into little pieces and showed that it would heal itself and all kinds of things like that. But now he's one of a very few labs working on this organism. He needs more people that will be working on it in order to help him and help everybody really make faster progress. So one of the disadvantages of choosing, the advantage of choosing a new model organism or something that's not been well enough studied, and there are huge numbers of possibilities of systems that have been only very poorly studied, not studied with modern techniques. But the disadvantage of that is that if you're the only person working on it, it's going to go very slowly. So, you know, some of you may know that in the very early days of molecular biology, Max Delbruck, a physicist, and others, his friends, a small group of people decided they'd focus on bacteriophage T4 and on E. coli. So there would be a critical mass of people that could interact. And I think that kind of thing is a very good idea. It could still be done today. For example, a university could hire a group of young faculty who want to work together and really work on a problem that nobody else is working with with modern techniques. I mean, it's just so many fascinating things that need to be understood, and it's really an art form. I mean, faculty can try to, as we're trying to do in our course, trying to get people to think more broadly and more ambitiously about science. And older people could help you think through this. And, you know, you could start by, you know, the easy thing to start with is a bunch of friends, maybe a bunch of postdocs at the same institution, or now you could do it online, so it doesn't have to be in the same place. Let's form a study group about, you know, trying to work on this problem in this organism and see what you as a group could come up with as ways of approaches. But that's the wonderful thing about science. There's just so many opportunities, and it really should be a very creative endeavor. I sometimes talk about, you know, make an analogy between being a really effective scientist and being a really good artist. And I talk about the fact that, so, you know, you could give me all the paints that Rembrandt had and all the materials he had, and I could do nothing that was interesting. it's not just the techniques you have and the things you can measure, it's how you put them together and to make a great piece of art, in the case of Rembrandt, or a great artist, or a real productive scientific enterprise. And that's a lot of the fun of science, is you have that challenge and that opportunity to create something that's new and important. So I think, it can't be done by a single individual working alone. It really needs to be, you need to stimulate each other. So, if I was young, and was in India, I would try to organize some groups of people, a group of people to, form a little study group with me by Zoom now. I don't have to be, it doesn't have to be in person. Explore new possibilities and get excited about coming up with some new strategy and for research that, in the end, more than one of them might end up setting a lab to work on.
Dr. Krishna Chaudhary.
Thank you very much, sir. And one more last questions for this session. I want to ask a question that you told us that PARP inhibitors are now being used to control tumor growth. My question is, why this approach is limited in some specific type of cells like breast and lung cancer? Why not in other type of cells?
Well, we've been doing cancer therapy for a long time. Some of it's very successful, but mostly what we do is, hit cells with toxic agent and hope that it kills the cancer cell more than it kills our normal cells. Agents that damage DNA, for example, are very effective in curing some cancers, certainly in slowing them down. But they're also you can't use too much of them because then you kill the normal cells. So the whole idea is, well, first of all, we now know from DNA sequencing, RNA sequencing, all those things, that every tumor is really different. There's a huge amount of heterogeneity. There's no such thing as a breast tumor. It's not breast cancer is many different diseases. There are different categories, but even within each of those categories, the cells are quite different. It's because the way they arose. Cancer, when I was teaching medical students, I used to teach them the cancer part of the medical school, cell biology. I said the really amazing thing is that all of us don't die from cancer before we're born, because we have billions of cells, and we know that the mutation rate, even with all the controls that we talk about in our book to create the, to repair DNA, to error correct DNA replication errors, and all those things that we do, we still have, an error rate in copying DNA that is significant. And so with billions of cells, every mutation is going to happen, in some cells somewhere in your body. And so, for example, if it's possible for, the change in one gene to cause a cell to grow out of control, you never even get to an embryo, because before you got the embryo, you'd have cancer. So as larger and larger multicellular organisms evolve, we had the part coming along with us all these methods to cause the cell to commit suicide if it's doing something wrong. For example, apoptosis, other mechanisms to check, should I really be growing, or, I'm in the right place. It's just amazing that all these checkpoints developed because they had to. You couldn't get a big organism otherwise. Cancer would have killed it really early. So the random process that gives rise to tumor, is a natural selection over many years, and it leads to lots of different ways of getting lacking the controls on growth that you need to prevent the tumor from killing you. So it's really not surprising that there's so many different ways to be a tumor. But then, of course, what we want to do is exploit. I would urge the people who are thinking about this to look up the PARP inhibitors in tumor. Alan Ashworth was one of the major scientists who developed this approach to cancer therapy maybe 10 years ago, 20 years ago, maybe it started. PARP inhibitors, poly-ADP ribose polymerase inhibitors, they're very efficiently in killing tumors that are hypermutable because of one particular type of mutation. Mutations in the so-called BRCA1 pathways. So, and you know, other tumors, they don't do anything. So that's that. If you study that example, and you could see the potential for, because almost every tumor is unusually unstable in its maintenance of DNA sequence, because that helped it to evolve. So in principle, you could do the same thing with many different tumors if you knew exactly why they were unstable. You'd make them hyper unstable with a treatment that would kill that cell, but it wouldn't be enough to kill the normal cells because they are already at a much greater fidelity than the tumor cells. So this is just one example of personalized medicine. It's just going to be much more effective in the long run, but it requires huge amounts of knowledge. And you know, I tell young people that not to work on cancer research unless you could find something that really is different from what everybody else is doing. There's just too many people doing the same thing, and most of the results, most of the research labs I see working on cancer research, I think have not thought carefully enough about finding a unique strategy that could really make a difference. I guess it's partly because it's easier to get money just by doing certain kinds of things that are fads, but you know, you have one life to live. You might as well do something that makes a difference.
Thank you very much, sir, for your valuable talk. Now the platform is over to Dr. Rupa Chakraborty.
So we are coming to the end of the program. Now I would request our head of the Department of Botany, who is also the joint convener of the program, Dr. Mithun Maji, to give a formal vote of thanks.
Hello, am I audible to all of you?
Yes.
Yes, audible.
Thank you, sir. Thank you, Honorable Professor Alberts, all the dignitaries, our most valued participants, ladies and gentlemen. It's a great privilege to propose a vote of thanks on this occasion. Today, my words are not enough to express gratitude. I, on behalf of Dinobandhu Andrews College, Department of Botany, would like to thank Professor Bruce Alberts, who graced us with his thought-provoking address on science, biology, and the world's future. We are very much thankful to you, sir. I must also thank Captain Hastings, ma'am, for her constant support whenever we have communicated, got your positive responses all the time. Thank you, Captain, ma'am. I would like to thank our principal, sir, Dr. Somnath Mukhopadhyay. Sir, we will remain grateful to you and thank you for being with us as a constant support. I may like to express gratitude to our IQC coordinator, Dr. Amitav Roy, for his constant encouragement. My heartfelt thanks goes out to all the teaching, non-teaching, and library staffs of Dinobandhu Andrews College, who always stand by us. We feel proud and thank you for making us feel happy with your presence to this webinar. I would like to take this opportunity to place on record A hearty thanks to all the viewers all over the world through the web platform, both in Zoom and YouTube. I must also mention the valuable technical supports from our departmental students. Once again, I want to state that we are very much grateful to you, Professor Alberts. Thank you for being with us. It's been a great pleasure to hear you, and it's a great honor for us. Thank you very much, sir. Thank you. Thank you all. Stay
Thank you, sir.
safe.
Thank you, sir.
Thank you, sir.
Thank you. Thank all of you for attending and asking such good questions. And I just want to emphasize, we need all you to be really good scientists because we got lots of problems to solve. And we need all of you to think about reaching out to your nation and other communities to spread science and the scientific temper throughout India and the world. So thank you very much.
Yes, sir. We will
Thank
definitely
you,
try
sir.
our best to
Well, sir.
take your comments, sir. Thank you, sir. Thank you, sir.
Thank you, sir, for being here. Thank you, Principal, sir. Thank you, Bruce, sir.
We wish you good health, sir.
Thank you.
Thank you.
Thank you.
I request our Principal, sir, to just officially close this program. Sir, the closing
Unmute-
statement.
Unmute yourself, sir. Sir, unmute
Principal,
yourself, sir.
sir, please. Principal, sir, please unmute yourself. Sir, yes.
It has been the greatest privilege for me to hear Professor Alberts, although in a virtual platform, and I congratulate all participants. That they have been able to listen to Professor Bruce Alberts in a nice way, and the message he has thrown to this virtual platform should be our pathway of life. So we will try to follow it for rest of our lives, sir. Thank you very much. Thank you very much.
Thank you.
This virtual webinar may be closed now.